In this episode, we're going to talk about malaria and the role of natural selection in combating it. Malaria has been a major agent of natural selection in humans. As noted previously, malaria was more widespread in the past. The effect of malaria on human evolution is also the first clear example of selection in humans detected, which is why you were assigned one of the original papers to read, that of Allison, 1954. Genetic resistance to malaria occurs through both modifications of the immune system that enhance immunity to this infection, and also by changes in human red blood cells that hinder the malaria parasite's ability to invade and replicate within these cells. There are probably many more genes involved than listed here. Also, keep in mind that different populations have different alleles of these genes, and many alleles have arisen multiple times independently. In this episode, we will focus on the G6PD gene, while the alpha and beta globin genes and the Duffy chemokine receptor gene will be the focus of the next. It might seem odd, but we will start by discussing the effects of eating fava beans, a food commonly eaten in the Mediterranean and parts of Africa that affects malarial resistance. Eating fava beans mimics the effect of changes at the G6PD gene that are also found in some people in the same areas. This is because fava beans produce an enzyme that affects the same pathway as G6PD in red blood cells. This pathway is the pentose phosphate pathway. It is a metabolic pathway that supplies reducing energy to cells, such as erythrocytes or red blood cells, by maintaining the level of the coenzyme nicotinamide adenine diphosphate, or NADPH. The NADPH in turn reacts with GSSG with the help of the enzyme glutathione reductase to produce glutathione, as well as NADP, which goes off to get another hydrogen. This maintenance of the level of reduced glutathione, or GSH, means that it is available to counter oxidative damage caused by hydrogen peroxide. While hydrogen peroxide will bleach your hair, it's not good in cells, so GSH protects the red blood cells against oxidative damage. The pentose phosphate pathway is the only source of GSH in red blood cells. The role of red blood cells as oxygen carriers puts them at substantial risk of damage from oxidizing free radicals, except for the protective effect of GSH. When all remaining GSH is consumed, enzymes and other proteins, including hemoglobin, are subsequently damaged by the oxidants, leading to electrolyte imbalance, cross-bonding, and protein deposition in the red cell membranes. Damaged red cells are phagocytosed and sequestered, in other words, taken out of circulation, in the spleen. Fava beans, also known as broad beans, affect one part of this pathway. Specifically, they contain high levels of visine, divisine, convisine, and isouramal, all of which are oxidants, and they lower the amount of GSH. This then increases the amount of oxidative stress in the red blood cell. Glucose 6-phosphate dehydrogenase, G6PD or G6PDH, is a cytosolic enzyme in the pentose phosphate pathway, and being deficient in the enzyme because of mutations in the gene results in greater oxidative damage, lower NADPH, and lower GSH. G6PD is present in all human cells, but it's particularly important to red blood cells, since mature red blood cells lack nuclei and cytoplasmic RNA. They cannot synthesize new enzyme molecules to replace genetically abnormal or aging ones. All proteins, including enzymes, have to last for the entire lifetime of the red blood cell, which is normally 120 days. When both G6PD deficiency and fava beans are present, you get even more reduction in glutathione reductase. People with G6PD deficiency are at risk of hemolytic anemia in states of oxidative stress. This will include eating fava beans. This is known as favism. Not everyone with G6PD deficiency will have a reaction to fava beans. In 1956, Alving and colleagues showed that in some African Americans, the anti-malarial drug primaquine induces hemolytic anemia, and that those individuals have an inherited deficiency of G6PD in erythrocytes. G6PD deficiency is common in Mediterranean, African, and other populations. G6PD deficient persons are also sensitive to several other drugs, in addition to primaquine. G6PD deficiency is a sex-linked disorder because the G6PD gene is found on the X chromosome. Specifically, it's classed as sex-linked recessive. Remember that females have 2 copies of the X chromosome, and males have only 1 copy of the X chromosome. They also have a copy of the Y chromosome, which is what makes them male. So that females don't have twice as much enzyme or other X chromosome gene products as males, 1 copy of the X chromosome is inactivated in each cell. This occurs randomly in every cell. For G6PD, the level of enzyme is classed into several categories, 2 of which are non-clinical, quote-unquote, normal levels. People with severe deficiency have less than 10% enzyme activity in their cells. These people fall into classes 1 and 2, depending on their symptoms. People with 10 to 60% enzyme activity may have mild anemia if exposed to chemical stressors, including those in certain drugs or fava beans. Here you can see 2 pedigrees or families. In the first, the father is affected by G6PD deficiency. The allele of G6PD that he has only produces the version of the enzyme with low activity. The mother is unaffected with both copies of the gene producing the enzyme with full activity. Their 4 children each receive 1 copy of the X chromosome from their mom. So in this case, it always has an allele with full enzyme activity. The sons get their Y chromosome from their dad, so they are unaffected by G6PD deficiency. The daughters get their father's X chromosome, which has the deficient allele. However, since they have 2 X chromosomes, they are likely to be unaffected or possibly mildly affected. In the second pedigree, the mother is an unaffected carrier and the father is unaffected. In their children, 1 son is unaffected and 1 son is affected. The daughters are either unaffected or unaffected carriers. Basically, it depends on which copy of the X chromosome the son or daughter receives from mom. Think about what kind of parents it would take to get an affected female. Do you expect females to be affected by severe G6PD deficiency as often as males? G6PD deficiency is the most common enzyme deficiency in humans, estimated to affect some 400 million people. There are many mutations at this locus, and some of the common alleles are shown on this map, which also shows the percentage of the population with G6PD deficiency. 2 of these alleles attain frequencies of 20% or greater in African and Mediterranean populations. These are termed the A- and MED mutations. Mutant varieties of G6PD can be more unstable than the naturally occurring enzyme, so that their activity declines more rapidly as red cells age. Whether G6PD has an effect on malarial impact has been studied in populations in Tanzania, East Africa, and the Gambia, West Africa, where anti-malarial drugs were not commonly used. These studies followed children during the period when they are most susceptible to falciparum malaria. In both cases, malarial parasite counts were significantly lower in G6PD deficient persons than in those with normal red cell enzymes. In addition, because the enzyme deficiency is sex-linked and female heterozygotes have random inactivation of an X chromosome in the progenitor cells that create red blood cells, a population of G6PD deficient red blood cells coexisting with normal red blood cells is created. In this case, malaria parasites were significantly more often observed in normal red cells than in enzyme-deficient ones. So, for G6PD, being slightly deficient appears to help protect you from malaria, but being too deficient puts you at risk for anemia. This is a classic example of balancing selection, where more than one allele is favored in the population. In other words, in a malarial environment, not being deficient puts you at a greater risk of dying from malaria, but being too deficient puts you at a great risk of dying from hemolytic anemia. Somewhere in the middle is best. An evolutionary genetic analysis of malarial selection on G6PD deficiency genes was used to examine the evolutionary history of G6PD and look at the timing of selection. In other words, when did these deficient alleles spread? Vorelli et al. Looked at sequence variation at G6PD in different populations. The red positions shown here affect G6PD production. For example, here you have several alleles that have a G at position 902 in the DNA sequence of the gene, and one of these was common in their sample from different African populations. All of the variants with the change at position 902 are the so-called A- alleles. Based on the amount of diversity in the Chi-Ping and Met alleles, and a constant mutation rate, the researchers concluded that these alleles likely arose in the last 5,000 years. The diversity found in the African A- alleles suggests that that variant arose within the past 4,000 to 11,000 years, but that the A allele, which is not deficient, is much older. In summary, newly mutated G6PD deficiency alleles arising in a malarial environment can quite rapidly become common within 1,000 to 3,000 years, depending on the intensity of selection. The rise of deficient alleles within the last 10,000 years is likely related to the spread of P. falciparum as land was cleared for agriculture. Studies of genome variation and evolution of P. falciparum suggest that this species expanded in the last 3,200 to 7,700 years. These dates coincide with the spread of agriculture, which increased the density of populations, forest clearing, and urbanization near sunlit pools of water. Such conditions favor the breeding of Anopheles mosquitoes and the transmission of malaria. While G6PD has been under balancing selection in human groups living in regions with endemic malaria, G6PD evolution overall in primates has been subject to purifying selection. In other words, most changes were not tolerated. Over the course of primate evolution, this gene has been highly conserved, which is apparent from the few mutations that cause amino acid change that we see. Here, the percentage amino acid change is shown above the branches of the phylogeny. This is not terribly surprising, given that G6PD is an important enzyme.